Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Tobias Schenk, PhD, Product Manager from Sartorius Dedham Biotech.
Thank you, Alex, for the kind introduction. Welcome, everyone, to the webinar, Shaken, Not Stirred. The question what we want to tackle during this webinar is really if rocking motion versus impeller technology, what is optimal for cell cultivation, which conditions do you need to consider, and for which application maybe the one or the other, or at the end maybe the combination is best for your cells. So first, I will provide a quick introduction into both methodologies. I will introduce briefly only the Biostat RM rocking motion product family so that you know what does it consist of, what does it need, and which parameters you can control. Same is true for our stirred bioreactor product family. And then I will talk about application data points to prove what is better rocking motion, what are their applications, data, what is better for STR. And then at the end, I will summarize this at the slide Shaken, Not Stirred. So let's start with an overview of the Sartorius bioreactor product portfolio. And as you can see here on this slide, it really ranges from small scale, if you have the need of process development, multi-parameter testing in our Ember scale, then it goes up to universal 2-liter. Then the rocking motion bioreactors, RM2050, 200 come into place. And if we talk about pilot or production scale, then obviously the large scale stirred tank bioreactors up to 2,000 liter are important. And in this webinar, I want to focus on how those instrumentations play together and where, in which application, which technology is best in its performance. So first, let me introduce a bit what is the rocking motion bioreactor technology, what is the benefit, how does the cells survive even in a rocked or in a waved bioreactor. The gassing is passive here, and also the mixing of the nutritions of the cells is introduced or induced by these wave motion, by this wave motion. And instead of actively gassing, instead of bringing nutritions actively or also gases actively into the cell culture media, this is done by the wave. The culture media is infused into FlexSafe bags, and they are available in different working volumes and is very sensitive to the cells. There are predefined protocols available, and it is similar to it's similar to kind of like a shake flask, but in motion, so that really the cells are during their cell cultivation in optimal state. The system consists of, as displayed here, are available in 3 different options, either the basic RM2050, which comes without a tower, control tower, or the RM200, which can handle liquids or cell culture media up to 100 liter as a working volume, or the smaller brother, the Biostat RM2050, up to 25 liter of working volume. So as you can see here, you can start very low with 500 milliliters to establish processes, but you can also go large scale up to 100 liter of working volumes. Compared to the stirred bioreactor and the technology there, as I explained already by the wave-introduced motion, here in contrary, we do have active stirring and we do have active gassing and that's the classical conventional stirred tank bioreactor design. We have those blades which evolve or help support that the nutrition and the cell culture media and the cells itself are distributed homogeneously. And also the gas is provided into the media actively due to the impellers. As you can see on that screen here, the distribution is very homogeneous, and this is true for all different conditions, no matter if you stir slowly, if you have a low working volume or high volumes, it delivers an outstanding mixing efficiency. And also here in the STR portfolio, you do have the possibility to upscale from the Amber small scale multi-parameter testing 250 milliliter up to 2000 liter in the pilot and production scale. It is a simplified technology, and it allows the transfer between the cultivation scales without bridging studies, because the same geometric scale and impellers used in the small scale is also used in the large scale. So that's definitely a benefit similar to the RM. Once you optimize your processes in the small scale, you can seamless upscale up to 100 liter. So let me briefly introduce the Rocking Motion product family. So what is the BioStat-RM system? It consists of 3 different components. The FlexSafe bags, that's the bag where the cells are stored and cultured with cell culture media. The FlexSafe bag with the medium and the cells is placed onto the rocker platform. The rocker platform can control the speed of rocking, the rocking angle. It is temperature controlled. So for example, that your cells always grow at 37 degrees. And it has also pH and DO levels for continuous monitoring of process parameters. It can also be added with advanced control systems, our BioPad sensors like biomass or trace. And the third component is the control tower. And the control tower is, so to say, the heart of the system, because it controls all the sensoric coming from the rocker to the tower. It enables automated and controlled batch run, fed batch, or even high cell density perfusion cultures. The working volume, as explained, is from 100 milliliter minimum working volume to 25 liters in our 2050 rocker, and it can go up to 100 liter in our 200 rocker. The tower is driven or is embedded with the software BioBrain. And this is also our Sartorius world, because if you as a customer are used to the BioBrain surface, the user interface, then you will have the same controls at the Rocking Motion as well as at the STR. And the control tower fulfills all the commercial manufacturing norms and regulations. It's validated for usage in a clean room environment, and it can be used in a GMP grade B environment together with the rocker and the FlexSafe bags. We constantly update our Rocking Motion as well as our portfolio for stirred. And here, this new developed control tower really is adapted to customer needs. For example, the pumps rotate in either directions, so you can use them for harvesting as well as for feeding, and they can be configured completely flexible and independently depending on the user needs. The tower is smaller than the previous one, and it can be stacked if lab space is limited. What are the typical applications for Rocking Motion systems? And here our largest system is displayed, the RM200. It is used in preclinical and clinical studies for fast protein and cell supply for commercial manufacturing. Then also large-scale seed expansion. I will talk in the application section a bit more about this. Small-scale production, if monoclonal antibodies or recombinant proteins need to be produced, not in large scale, but small scale, 100 liter is sufficient for this. and it can be all our RM systems can be used for GMP production, not only the 200 system as displayed here. And briefly, I want to talk about the FlexSafe RM bags, since it is a crucial point in our RM system. With the basic bag design, no pH and DO control is possible. It's really just cultivation under constant conditions. If pH and DO need to be constantly monitored, our optical bags are a perfect match. And for perfusion processes, we have specifically designed perfusion bags with a fixed membrane in it to really allow those perfusion processes done on the rocking motion platform. All our bags can be customized and have also access, or the availability of biomass sensors is given. Quickly, because perfusion is a high topic for our customers, our perfusion bags have bound or a sealed perfusion membrane on the bottom of the bag, and this is fixed. And with this, it is optimized for, or suitable for, even the most sensitive cell lines, because the perfusion membrane is not swimming in the cell culture membrane and sometimes even pushing against the bag walls, which could add additional shear stress to the cells. So that's why our technology is patented exactly for these purposes. Then a quick introduction into the SDR product family. We offer here as well from 12.5 liter up to 2000 liter a seamless scalability. The single-use bioreactor is engineered around a conventional stirred tank bioreactor design. I think that's no new information here. And the synchronized hardware between the BioBrain software to control the stirred bioreactor, the bag holder with integrated temperature gassing and sensor control, and the FlexSafe SDR bag. That's crucial, and that are the key components how the concept plays together and then can be used as a stirred bioreactor tank for cell culture processes. With our generation 3, we have a new control tower design, which is independent from the bioreactor size. It's always the same size, no matter which stirred bioreactor tank you are controlling with it. It is able to support standalone GMP installation. We fulfill the required industrial standards with our SDR generation 3, and we have the option to integrate load cells into our stirred bioreactors so that also the weight can be controlled during process. Here, very important, the FlexSafe film is the same in our SDR bags as in the RM bags, as well as in our storage or shipping bags, which really is crucial for an excellent cell growth. It has a superior robustness, and it really helps the lot-to-lot consistency of cell growth, robustness, purity, sterility, and cleanliness. So that's an outlook why FlexSafe is used in both technology. And furthermore, to enable even intensified processes, you have also with SDR the possibility to add additional sensors to the FlexSafe bag. For example, the BioPad biomass feed, biomass, where you can control even the feed or the bleed into your cell culture, or the BioPad trace, which allows to monitor the glucose concentration to maintain a healthy metabolic state during the cultivation. And also here, customized SDR FlexSafe bag designs are available. But now to the most important part, I guess, of this webinar, really applications and data to show which application are using rocking motion technology and which applications are using SDR. So here, what you can see is the cultivation results obtained from Dr. Eibel. It is published by an application report for HAC 293 cell lines. And you see here a static system, small scale shake flask, but then the two middle section biostat RM20, that's rocking motion. And in the last line, biostat B universal glass, that's stirred motion bioreactor. And what you can see here is that the viable cell density reached is very similar levels. Also, the doubling time is similar or even the same, no matter if you use rocking motion technology or stirred motion technology, and even the maximum product titer is the same. But the most important difference here is with the stirred motion universal, it's a multi-use equipment versus rocking motion single-use flex-safe bags. And with the multi-use, you have the downtime due to cleaning, due to sterilization. You need to set up the whole process. With the rocking motion single-use flex-safe bags, it comes pre-sterilized out of the box. You can use it right away, and it is very easy to use and to connect to the lines to start the rocking motion, and you can begin your cell culture. So this really shows that even for HEC 293, you can use both technologies, but from a handling perspective, there is a difference between multi-use and single-use. On this slide, though, I want to share data for high density N-1 perfusion culture, where we used rocking motion in combination with stirred technology, and we even utilized an automated inoculation of both bioreactor types. So what do you see? On the left side, you see the rocking motion perfusion seed culture. The data shown here is only for the 50 liter, but the experiments were done in all different RM scales, 2 liter, 10 liter, 20, and 50. And all bioreactors, rocking motion bioreactors, reached 100 million CHO cells per milliliter in less than 7 days by maintaining the viability above 90%. When those 100 million set point was reached, due to the biomass sensor in that flex-safe bag, we could activate the automatic inoculation so that the stirred bioreactor next door was automatically inoculated with the cells from the rocking motion, and then started directly the perfusion process in an SDR 200 liter bioreactor. And with this, you could see comparable viable cell densities and the viability observed during the perfusion process stayed the same with manual or with automated inoculation. So in this case, the key message really is sometimes is less the question of should I take on the rocking motion technology or should I decide to go with the SDR? Sometimes the combination brings benefits and can also help to intensify certain processes. And I have also some slides for the intensified process later on. But let's take a step back and again compare the outcome and the cell culture parameters between rocking motion and stirred bioreactors. And here the case study shows and compares data from the 50-liter biostat rocking motion and the 200-liter biostat rocking motion with the 50-liter SDR and 1,000-liter SDR. And what you can see here in yellow are the results, no matter the viable cell count, no matter if it was being counted in the rocking motion or in the stirred, it stayed during the time of 17 days in the same range. Same is true for the viability. It stayed in the same range, and as well for the product, the harvested product at the end of the process. So we can prove the viability and the performance even from small scale, so from process development up to large scale, up to commercial scale. Once you set up the process, you can upscale and expect the same results, even from small scale SDR to large scale, or even from RM to SDR. Then I want to point out to this publication from 2016, because it really tackles the topic of microcarrier cell culture. So stromal or stem cells which need to or which are pending on microcarriers in the culture. That's a typical application for stirred bioreactors because not many people tested this in rocking motion. One argument, for example, was that in the stirred bioreactor, there's a good model how to mimic or how to predict the fluid velocity in the stirred bioreactor, but it was not yet available in rocking motion. But with this comparison here in the middle, you see the fluidic model in rocking motion bioreactors, depending on the filling volume, 500 milliliter on the top, 1 liter in the middle, and 1.5 liter in the bottom. And we can exactly predict and compare the fluidic velocity, the liquid flow, filling level, the rocking rate, and the angle to minimize the buildup of quality influencing microcarrier cell aggregates. And in this publication, it was very nicely written that even those stromal or stem cells, depending on microcarrier cultures, it can be as well cultured and predicted in rocking motion versus stirred bioreactor. So rich cells mainly then can be cultivated in the Biosat RM systems, and we can talk about our experience for so many times in the field. Shear stress sensitive cells, such as stem cells, immune cells, for example, is a typical application for rocking motion reactors. Mammalian, some insect cells are also used, and as a result of all this, the molecules listed here are all relevant and are all produced and used from customers in our RM portfolio, like vaccines, cell and gene therapy, biosimilars, and MAPs. Some of them use it for C-train, some others use it for small-scale production. And as I pointed out earlier, sometimes the question is not only is it either RM or STR, sometimes the combination provides you a higher benefit than if you would go only for one of these technologies. I think most of you are familiar with the classical fed batch, also with some intensified strategies. And I want to pinpoint here how RM and STR can play together to really deliver a benefit to you. Classical fed batch, typical 3 to 4 weeks. I would say we all know, I guess, that you start from a very small cell number, you thaw the cells, you inoculate a very small rocking motion bioreactor, then you go to the next scale, RM200, so that you have enough cells to inoculate a stirred bioreactor, let's say 500, and then you go finally to the large scale 2000 liter. Typically done for over years, very conventional. And nowadays, a lot of people talk about intensified processes, and there are two different possibilities. Either you intensify with a stirred tank so that you go directly from the rocking motion to stirred 500, and then you inoculate the 200 at the 2000. With this, you can increase the titer for some clones, and you reduce the culture length. Or on the right side, you use the more economic scale that you inoculate from the RM50 to the RM200, and then from the 200 directly into the 2000 STR. With this, you lower the cost of the consumables because the RM or the rocking motion bioreactor is more economic in maintaining and usage, but you maintain your titer. And with this, I'm already at the summary slide. What should you go now for? Is it shaken or is it stirred? With this slide, I would provide kind of like the summary and the overview. I mean, stirred motion technology has been the gold standard for a long time. If you go for large scale up to 2000 liter, that's definitely the way to go. It depends on the cell type you have. If it's a shear sensitive cell line like CAR T cells, stem cells, and then rocking motion has definitely an advantage. And I think also in the application data and the slides I showed before, I could prove that rocking motion technology is an excellent alternative for shear stress sensitive cells. And in some cases, the combination of both technologies, especially when we talk about intensified C-trains, is the key to go. If you want to have cooling, then this is not available at rocking motion, but in stirred. And the complexity, the setup of the different technologies is definitely more simple in rocking motion. although a stirred bioreactor is also not very complicated to set up. So with this slide, I want to leave the webinar and to leave to you if you have some questions for this, and you could use this slide as a guidance if in future you need to decide which technology you should go for your cell culture process. Thank you, and I would open up for questions.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, would it make sense to use small rockers for NX perfusion within seed train? You mentioned just N1 perfusion.
Yes, that's a very good question because it's true. I focused on N-1 seed train. It is absolutely true. You can expand the seed train to even more stages, N-2, N-3. And if you remember the slide, what I showed before, where the economic intensified seed train consists of two different rockers, you can play this even with two different bags. So you can use the RM2050 and you start with a 20-liter bag, then you go to a 50-liter bag, let's say, and then you inoculate the 200 RM. With this, you always stay within kind of like the economic scale of the rocking motion technology, and only at the end when volume really matters, if you need then to go to 2,000 liters, then you use the medium and the cells from the large scale rocking motion to inoculate the stirred. So yes, you can absolutely expand what I said also from N-1 to N-X seed train.
And can you describe shear stress or gas supply quantitatively in your rocking motion stirring bioreactors?
Yes, because the gas supply is actively done in stirred, right? So you will have some gas bubbles, air bubbles, let's say, and especially when they pop at the surface, that can add some stress, and it disrupts a little bit the homogeneous distribution of the cell culture media, as well as the stirrer will impact some shear to the cells, depends on how fast you stir. And with the rocking motion, wave-induced, what I showed at the very beginning, the slide, there you don't have any active gassing, meaning you don't have any gases or gas bubbles which could pop at the surface. So this is less, or this causes less shear stress, and as well the rocking motion. Again, here depending on the speed you apply to the rocker, it is less harmful to the cells. At least when we see this also in publications, for example, immune cells mainly are cultivated in rocking motion versus stirred bioreactor tanks so far.
And could the cells be impacted by the transfer from RM to SRT culture?
Excellent question, because you need to transfer them, by the way, also from one STR to another if you upscale, if you need a larger scale, and that impacts the cells not much. At least the transfer from one STR to another is the same as you would transfer from RM to STR, and our tubes are large enough, our pumps, you can vary how fast and how rigid the pumps should transfer the cells from one bioreactor to another. So if shear stress then is a really sensitive topic for your cells, then I advise to use a lower speed of the pump. That's beneficial then for the cells when you transfer them from one bioreactor to another, but there's not more and not less impact to the cells independently if you do it from an RM to a SCR or from two RMs.
Okay, and our next question says, thank you for a great presentation. How do you regulate and assure proper gassing oxygen levels and to control pH, etc. in RM reactors?
Very good question, because yeah, obviously, if you compare both technologies, you would assume, well, if I don't do it actively, how do I control, right? How do I monitor? How to assure that the cells always have the best, the best conditions? So in the rocking motion reactors, you do have the possibility to control the pH due our optical sensors. You can actively also influence the pH by adding ingredients to control the pH, or, and this is what most customers are doing, you do it via the cell culture media. So you add always a bit more new media, or you do you exchange the media completely if we talk about a shot-based perfusion, and the media itself has some pH buffering conditions. And if you see, and that's the reason why, by the way, you also control and monitor dissolved oxygen and the pH, if you see that the values are below or above your threshold, what you believe it's best for your cell culture, you take action and either increase the shaking to bring more oxygen into the cell culture media, or you exchange completely the media if you believe that the pH needs to be adjusted, or you actively adjust the pH via ingredients. And we have ports and assemblies on each FlexSafe RM bag, which enables you to do so.
Okay, so it looks like we're running a bit short on time. If we don't get to your question, we'll be passing them along to Tobias, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, in the last slide, if I understand well, you mentioned there is no temperature reduction for rocking motion bioreactors. Could you explain this point in detail?
Yeah, maybe that's a good last question indeed, because when I talked about cooling options so that we can actively reduce the temperature, currently that's not possible with our current rocking motion bioreactors, because it doesn't have active cooling. We have only active heating. Our stirred bioreactors, they have both technologies, cooling and heating embedded. But indeed, this is evaluated internally because in some applications, it is of high interest that you cool the cells down prior to harvest. So that's why if you're looking currently for a cooling option, or you have also cell cultures which require lower temperatures, lower than room temperature, then the STR is currently the technology to go.
Okay, well, thank you, Tobias. And thank you to our audience. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with the direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.